So I changed the title section on this one because now we're going to bring all this into where the cancers begin to actually, what kinds of genes they affect. And so typically what is being affected are the growth regulating genes. If a cancer is an uncontrolled growth, then you are affecting things that control growth in a cell. So let's back up a little bit. Role of mitosis, two things. Initial growth of an organism. So you start off as a zygote, as a single cell, you need to get to a multicellular adult. Mitosis is what will allow us to get from the zygote to adult. Some cells will stay the way they are your entire life. Your nervous system, your nerve cells, once you get to your adult stage, that's all the nerve cells you get, which is why damaging them can be very detrimental. But some cells need to be replaced over time because they get a lot of wear and tear on them. And the group of cells that usually needs to get replaced a lot are epithelial cells. So epithelial cells are undergoing lots and lots of mitosis throughout the course of your lifetime. So if a mutation develops within the epithelial cells, then you have a much better chance of getting a cancer. And because they're constantly undergoing mitosis, you just have more and more chance over time in order for those things to develop. These are things like your skin, your blood vessels, the outsides of your organs. These are all epithelial cells. So, what can happen here? And this is what we're going to bring back in the cell cycle that we talked about. So, you have genes that control growth. So, these are cell growth genes. You can either have mutations occur in these genes. And if you do, you have something that regulates going to a non-regulator. So, if, for instance, you have normal growth here, and you disrupt one of these genes, you can now yield uncontrolled growth. So this mutation, it can come about by many different ways. It can be spontaneous errors within that gene as it replicates. It can be spontaneous. It can be induced. Radiation can cause this. Chemicals can cause this. Tautogenic shifts. Analog base pairs. Any of these things that can cause mutations specifically within this set of genes can lead to a problem. So, where do these genes come into play in the cell cycle? So you'll see figure 12.5 in your book. This is also in chapter 2 when we had our review. Most of your existence is interphase, or I should say most of the cell's existence is interphase. And then a very short period of time is what's called the mitotic stage. And the mitotic is mitosis and cytokinesis. So there are 3 genes that produce chemicals that tell the cell it needs to check what's going on at different places. One of these is the G1S checkpoint. So you will have a chemical that is produced either by the brain and sent to the cell, or sometimes these are made inside the cells. But a chemical will come to the cell and it'll do what is called signal transduction. And these are the proteins created by growth genes. So one place you want to check is the G1S phase. Once you have finished cytokinesis, you go into the G1 phase of the cell cycle. At this point, you may be needed or you may not be needed. At this point, this will make two decisions. At the G1S stage, it'll either say, we're not needed right now, so we're going to go into a stasis phase called G0. A lot of your blood cells will go into this. You'll keep making blood cells, but if you don't need any more at a particular time, it'll just say, we're going to put these on reserve. If, however, you need more, then this cell will undergo the synthesis stage. So this is why it's called the G1S. The signal transduction occurs during G1 and either tells you to go into G0, stasis, or move on to S when we synthesize our sister chromatids. A second place that will regulate this cell growth is at the G2. We call it G2M. I usually just call it G2. At the G2M stage, we're going to check, we're going to say, did we replicate that DNA correctly? So we're going to spot check the DNA synthesis. If the DNA was synthesized correctly, so if it's done correctly, we're going to go ahead and we're going to allow you to go to mitosis. However, if you don't synthesize it correctly, if it's an incorrect synthesis and we haven't repaired it, we're going to undergo apoptosis, that programmed cell death. If I've got bad copies of the DNA, one of the ways that your body checks itself is right here, and it says, hey, we didn't do a good job of this. This isn't something we want to pass on. We will undergo apoptosis. So hopefully, only if you synthesize your sister chromatids correctly will you actually enter the mitotic stage that will allow you to create a new cell. Because if you have a bad copy, this is one of the ways we actually catch mutations. There's something wrong with this. Let's just go ahead and get it out. So this is why there are lots of mutations that are happening constantly in your body. A lot of times, though, we get rid of that cell right here and we don't keep correcting it. We don't keep creating new lines. And then our last checkpoint, or our next checkpoint, is called the M checkpoint. At the M checkpoint, we make sure that we have actually set up those spindle fibers so that we divide the genetic information evenly. So remember, during the M stage, we have our sister chromatids. They are lined up on that metaphase plate. Remember, that metaphase plate is sort of this hypothetical line running through the center. We have our centrioles over here and over here. And remember, these centrioles are going to put out spindle fibers. This one should put out 4, so they can pull 4 sister chromatids. This one should pull 4, so that it will pull 4 sister chromatids. We will get an even split of the genetic material. So we come in and we say, hey, have we lined things up? If we notice for some reason that maybe this spindle fiber is now attached to this, that's an incorrect way of splitting it up. We're not going to get an even division. We're going to end up with maybe 5 chromosomes on this side and 3 chromosomes on this side. If that's there, then this will lead to, once again, apoptosis of the cell. We don't want to make bad copies of ourselves. So if we have these spindle fibers not making their correct alignment, they will undergo programmed cell death. If, however, each of these spindle fibers is aligned and it has the proper number of chromosomes on either side so that during anaphase we get an even division, this checkpoint will allow us to complete mitosis and undergo cytokinesis. So then we can go back through our stage again. However, if you interrupt or do something to these proteins, remember, genes code for proteins. If any of these proteins are mutated, you're not going to get the right signal transition. If this protein doesn't do its right check, we're going to keep making bad copies of bad cells, right? So if any of these three proteins that will be transduced to tell the cell to keep going or to stop, if any of them are compromised, we may not stop. We may not stop. We may just continue getting uncontrolled cell growth.